So please welcome Michael Shamash from McGill University.
Thank you, everyone. Just look at the charger. Okay, great. Good afternoon, everyone, and thank you all for coming to the phone session, and thank you to the organizers, especially for inviting me to the session to present a bit about what I've been working on at McGill. Today, I'll be speaking about two different aspects of experiential learning that I've been able to be a part of in our department, which is the Department of Microbiology and Immunology at McGill. And just to introduce myself, I'm a PhD candidate in the lab of Dr. Maurice. So without further ado, I'll jump right in. And I figured I would, as a first slide, just put up here a screenshot or a couple screenshots of some of the required and complementary courses that are prescribed to students taking the microbiology and immunology major in our department. And of course, a lot of the courses make sense for what you expect for a microbiology degree, basic genetics at the top, some virology courses, microbiology lab, parasitology, immunology courses as well. But one thing that really stood out to me as someone who went through this curriculum several years ago, I lost track, but also who is constantly TAing students who are going through this curriculum now, is that there's an overall lack of any kind of computational training when it comes to bioinformatics. And more, of course, I guess I could just transition right away, more and more bioinformatics is becoming a part of the research that we do. There's a lot more omics work being done, whether it's sequencing or metabolomics, transcriptomics, things like that. And we're constantly dealing with these big data sets that maybe the undergrads that are graduating now from these undergraduate programs don't have kind of the full skill set to be able to tackle on day one of, or the first month of their grad school studies. So just to briefly kind of review what bioinformatics is, it's the field of using computational tools and techniques to analyze any kind of biological data. And as I mentioned, it's becoming increasingly embedded across the life sciences. But ultimately, incorporating bioinformatics lessons into existing curriculum can be challenging for a variety of reasons, not only limited to time constraints or diversity of backgrounds, but also limited financial resources at the departmental level. So in collaboration with Assistant Professor of Teaching, Dr. Jasmine Shahal, and another graduate student in our department, Laura Melena, we developed a series of optional hands-on introductory bioinformatics workshops for undergrads in our department. So really, if they were interested, they'd be able to sign up and learn a bit about what bioinformatics is and how they could go about it. And the workshops really focus on the more practical aspects of applying bioinformatics in microbiology, using real-world data sets to answer meaningful biological questions, whether from a clinical kind of perspective or more of a biomedical research perspective. So when it comes to making kind of optional workshop, you got to get the word out, right? You need people to register since it's not a required course in the department. And when it came to advertising for the workshops, we advertised via departmental and student association newsletters, as well as on social media through the undergrad student association and just professor lecture slides. And these were the two kind of ads that I made for the workshops. We had two different kinds of workshops that we were running. The first was a strictly kind of dry lab workshop. So, and I'll get into what we did each one shortly, but this had, of course, what we were hoping to get out of the workshop, contact email if you had more questions, QR code to register, and the date and time options. And the second workshop was more wet lab focused, or had a wet lab and dry lab component, so same kind of information. And we limited registration to third and fourth year undergrad students just due to the necessary prerequisite knowledge, since a lot of the topics we were discussing kind of require that they already took like a basic genetics course or a micro course or something like that. In terms of the curriculum for these workshops, we prepared handouts, which were digital or printed if the student didn't have access to a computer, and slides for each of the workshops, which included learning objectives, the necessary theory for them to kind of review if they wanted or review after the fact, the protocols they'd be following, either in the lab or computationally, and any code they might need to reference or use, and any other links that they wanted to, or any other links that they could information. And each workshop was 4 hours in length, typically split over 2 evenings. We chose evenings just to accommodate everyone's schedules because, of course, all of the courses are scheduled at different times during the week, so this was easier to access registration and participate. So I mentioned that we had these 2 workshops that we were running, and each one we ran a couple times. I'll focus on the first one first, which had the overall kind of objective of using bioinformatics to assemble the genome of a mystery microbe from raw sequencing data and determine its identity. So in this workshop, students were assigned user accounts on a virtual machine, which is like a remote server that they could all log into and interact using the command line and bash scripting. So really interacting with the command line, interacting with the computer or the server using only their keyboard. And that for most students was a first for them. They didn't even maybe know that you need to interact with the computer in such a way because we're all used to using our mouse and just very visual environment most of the time. And I provided a curated Illumina data set that we had from our lab just to all these students so that they could work with that data, and all of the necessary tools were already pre-installed just to save time and we could hit the ground running kind of thing. And I'll mention now that this mystery microbe, and the secret stays in this room, is bacteriophage lambda. So we purposely chose any phage, but lambda is kind of one of the few classic phages out there, just because it has a small genome, and when we're doing all of our computational analysis, it should run pretty quickly. So a 4-hour workshop, we'd be able to get from start to finish quite reasonably. If we chose a eukaryote or fungi or eukaryotes, but any kind of, like let's say a bacterial genome, that might take longer to run, and we wouldn't be able to necessarily finish right now. Right, so for this first workshop, as I mentioned, it was split over 2 days. Day 1 was really theoretical content heavy. So this 2-hour session focused on the theory of DNA sequencing technologies with some pros and cons of each and when you might want to use each one. We discussed library preparation for whole genome sequencing. That is a big kind of black box that I've noticed in the curriculum, going through it myself years ago. They always say, okay, you get the DNA and then you sequence it. And this is how the sequencing is done, right? Whether it's Illumina or Nanopore or PacBio. But when I started doing sequencing myself, I realized, oh, there's like a pretty important step in between. So we discussed library prep, the different type of files that you would work with as a bioinformatician, whether it's FASTQ file from the sequencer, FASTA files, and how you'd interpret the quality metrics of each of these files. And we went over the pipeline that we'll be using in the next class or the next workshop session on how they would analyze the data. And to try and keep things, oh, I guess it's on another slide, but we tried to keep things engaging by having some hands-on, whether it was with actual flow cells that we were passing around from these different sequencers, or just kind of Q&A. So day 2, this was really the hands-on lab, and as I mentioned, this is the dry lab workshop where the students first had a crash course in using Bash and interacting with the command line. So just kind of some of the basic commands that you'll need to get around and how you can run scripts and things like that. And then they analyze the sequencing data that we gave them. So to try and keep it interesting for them, both of our workshops had a kind of mission slide, so a goal that they are trying to achieve by the end of the workshop. And this first workshop, your mission was to, well, after months of hard work, you and your colleagues have discovered a mysterious microbe in your lab. And now before you fully characterize it, you want to know whether or not it's novel. And if it's not novel, you should report back to your team as to what you found so that you don't waste your time characterizing something that we know already. And the DNA I mentioned was already extracted, and we did the sequencing ourselves on the McGill platform. So they just got this data set back and are ready to work with it. And this is the pipeline that the students used. So we gave them this raw sequencing data straight off the Illumina sequencer, and then we went through really the whole pipeline from start to finish, discussing and giving the opportunity to make these kind of quality plots with FastQC and multi-QC to check the quality of the sequencing data, using FastP to filter and trim their sequencing data according to the quality metrics that they gathered in the first step. Mega hit for genome assembly and NCBI's BLAST-M, for microbe identification. And here's just a photo of phage lambda case. So yeah, here are some photos from the first workshop. that Dr. Shahal took. And these are the flow cells that we were passing around. So for the flow cells, I just emailed the McGill Genome Center asking if they had any old unused or old used whatever Illumina flow cells that they would otherwise just throw away. And sure enough, they had a couple that we could use and just pass around so the students could actually see what an Illumina flow cell looked like. It was also my first time seeing an Illumina flow cell, which was pretty exciting. Highly recommended. Up in the corner, it's a bit hard to see with the resolution, but we have some nanopore flow cells as well that we got from our McGill. And as I mentioned, we printed some handouts for students who didn't have their own laptops. This is myself and Laura. And yeah, all of the participants had name tags as well, just to kind of try to make them more interested and more engaged and feel a part of this workshop, that they were actually doing something. And both, I think we ran 3 or 4 sessions of this kind of first workshop format, and pretty much all of them were full, so about 20 students in each, which was great to see. So just switching gears now to the second workshop. This workshop had, I guess I'll go right in, more of a wet lab component to it as well, where the students would first conduct the actual library prep themselves, do the DNA sequencing, and then come back 2 days later to do the genome assembly and identify the closest known relative of a, this time, pretty definitively novel microbe. So we actually didn't know what they were sequencing this time, just that it was a phage, because we have the SEA-PHAGES program that I'll chat about in a few minutes. So the sequencing that we used, the technology was using the nanopore MinION sequencer, just because it's very small, it's portable, and you can kind of do it all in a lab. You don't have to send stuff and wait weeks or months for results. So we can do it within the span of a week, no problem. Already, but students each had a different sample as well, which was pretty interesting. So they could compare their results with each other at the end of the 4-hour workshop. And we also received funding from the Gardner Foundation and the Fonds de Recherche de Québec, which is like the provincial version of the NSERC or CIHR, that made it possible to get the necessary equipment or reagents for this workshop, since it wasn't funded by the department. So the first day of this workshop was a balance of theoretical content and wet lab activities. We reviewed lab safety guidelines briefly, just because they are in a lab setting for this workshop, although most of them have already been in lab settings for their other undergrad courses. And we had a brief overview of the nanopore sequencing technology itself. And then the students actually did the library prep themselves using the rapid barcoding kit from Nanopore before I loaded the samples onto the MinION sequencer. And then they came back 2 days later with the FASTQ files ready for them, where we quickly again reviewed some of the theory of bioinformatics, whether it's interpreting quality metrics or these different kinds of files that they'll be using. But this time we did not use the command line environment, and we actually used a private Galaxy web server that I set up for the analysis of the sequencing data. And I'll get into that in a moment. Once again, there was a mission slide very similar to the previous set of workshops, except now they've discovered a bacteriophage from a soil sample, and their goal is to figure out its closest known relative and if it's not, which I think they pretty much all were. And the bioinformatics pipeline was very similar as well to the previous time, just different tools because we're working with long read data now instead of short read data. So quality checks with PycoQC or PycoQC, depending on how you pronounce it. Fly for genome assembly. We visualize the assembly using Bandage. I'm not sure how many of you have used this tool before, but it's a nice way to visualize your assembly and see how complete or incomplete your contigs might be, or how deeply you've sequenced and if it was even deep enough. But most of the time, since these were isolates, the assembly was really just one contig, so nothing too interesting to look at. And then we did a blast N at the end to identify the relatives. Yeah, so as I mentioned, we used a Galaxy web server for the bioinformatics analysis in this workshop. And the reason we chose to go down this route was because of, well, mostly time constraints. We didn't have time to necessarily do that crash course in using Bash and interacting with the command line. So instead, I chose to go a route that might be easier for some students to work with because we're used to clicking and dragging and all of that already in our daily lives. So this Galaxy web server I set up specifically for the workshops, and I installed the tools that they would need. There was a tab here, MIM workshops. That's an acronym for Microbiology and Immunology. When they would find all the tools that they would need for this workshop, they could just click and then upload their sequencing data and move the pipeline that way and download the outputs as they need, or just visualize them. So we saved some time there, and they can still get the same. I have a couple short videos. This was the wet lab environment, so it's actually the same lab that we run SEA-PHAGES in. Yeah, just to show you, each student basically had their own bench space that they could work at, and some of them were working in pairs if they had a couple samples each. And we used a mini PCR thermocycler, so it's surprisingly small thermocycler that can hold, I think, 16 to 20 samples. And you essentially program the cycles either through your phone or through the USB cable connected to a laptop, so there's Bluetooth as well. And yeah, that's where the students were loading on their DNA samples for the actual library prep protocol to take place. Instrument. This is the MinION sequencer itself, hooked up to the laptop that we were going to use for sequencing. We took this just before the sequencing run started. And then here I am loading the final library pool that's been cleaned up onto the MinION, and all the students are around watching, very interesting. So after both workshops, whether it was the first one or the second one that we ran, we conducted these anonymous post-workshop surveys using Google Forms just to get a sense of what the students got out of it and what they would change or what they were most interested in. And we provided time at the end of each workshop for them to complete this. They were given questions to evaluate the workshop's duration, the quality of instruction, the quality of the theoretical or hands-on content, and also text boxes to provide any other feedback or ideas that they had. And we saw that 41 out of 66 participants, so about 2/3, completed this survey, which is great to see. It's a pretty high number. And for most students, as I mentioned, this was really their first exposure to bioinformatics and DNA sequencing outside of the lecture hall, which was really exciting as well. We saw almost equal number of third and fourth year students. So McGill's naming of the undergraduate levels is a bit confusing, but U2 and U3 corresponds to third and fourth year. There was one freshman student who was really eager and said, sure, you can register, but you may not be able to get everything out of it, but we're happy to kind of support you along the way. And overall, the workshop duration was reported to be adequate, so the 4 hours seemed to be enough. On a scale of 1 to 5, most rated it at 3, so not too short, not too long. That's the middle. The different colors correspond to whether it was the first or second workshop, kind of the dry lab or wet lab one, but most said it was adequate. And in terms of the theoretical hands-on content, it was sufficiently advanced. So once again, at a 3 on 5 for most students, not too simple, not too complex. And same with the hands-on content as well. In terms of general feedback from the students, we saw that for most of them, they said that this hands-on aspect really improved their learning because they could engage with the content more and see how what they've learned in class can be applied clinically or in a research setting, since they were doing these experiments firsthand. And they also had great ideas for future workshops, some of which that I'm not expert on, but I'm happy to speak with other grad students to kind of get them involved and running these other workshops, whether it's microbiome analysis, population phylogenetics, or gene expression analysis, so I guess more RNA-seq kind of work. And that's it for the workshops. And I put back the title slide. I didn't know what else to put in between the two halves of my talk, but the second half of my talk will focus on another aspect of experiential learning that I'm involved in at the department, not at all related to bioinformatics or workshops, but actually related to phages, or bacteriophages, which, as you probably already know, are viruses that infect bacteria and are the most abundant, and I think the most, or definitely one of the most diverse organisms on Earth. They have all kinds of different shapes and sizes, high genomic diversity as well. And in terms of research, we've been able to get really great tools out of phages, whether it's the CRISPR-Cas system or DNA polymerases that have all been discovered or characterized by studying phages. Phages are now being used clinically for antimicrobial therapies and in the food industry, for example, or other research settings. But ultimately, the databases that exist out there for phages are heavily biased and limited in terms of their scope. So because of this, we need to study phages further, and we should characterize more phages from across the world in many different environments and habitats and so So this brings me to the SEA-PHAGES program that we mentioned already a couple of times in the previous talk and also in my talk. And of course, SEA-PHAGES is an acronym, as are most things in science, and it stands for the Science Education Alliance, Phage Hunters Advancing Genomics and Evolutionary Science. This is a program that was started in 2008 by the Howard Hughes Medical Institute in collaboration with Dr. Graham Hatfield from the University of Pittsburgh. And it's essentially a course, like a pure course, so of course for undergraduate research experience that takes place over 2 semesters, so usually a full academic year, where students go from an environmental sample, usually a soil sample, but some have brought in bark or water samples and things like that, all the way to isolating a phage, characterizing it, and then sequencing it and looking at its genome in the next semester. So at McGill, this is the setup we have, where in the fall, 3-credit course, it's a wet lab course, focused on the phage discovery component and isolation. And then in the winter, we have the genome annotation section, also a 3-credit dry lab course that students can take if they would like to continue. Now, originally, this program as a whole focused on one specific bacterial isolate, which was Mycobacterium smegmatis, which is actually one of the safe relatives of Mycobacterium tuberculosis. So there was a big interest, of course, in culturing phages that could maybe treat TB and of course, a wide variety of other bacterial infections as well. So there was a focus on M. smeg originally, but now they've added many other phages, or many other hosts, to the program from Gorgonia, Arthobacter, and Microbacterium genera, and some others as well. And just to give you a sense of some numbers as well, so to date, over 200 institutions across the world and 5,000 students have participated. At McGill, that's over 100 students since we started the program in the COVID year of 2020. In terms of number of phages discovered, over 26,000 phages were isolated. This screenshot was taken about a week ago. And 5,000 of those phages had their genomes fully sequenced. And over 86% of those phages had their genomes fully annotated and submitted to RefSeq or GenBank as well. So the high quality data that comes out of the program is added to public databases for other researchers to study or look on what they look like. This is our SEA-PHAGES team at McGill. So in terms of the wet lab side of things, it's myself and my research supervisor, Dr. Greg Maurice, who run most of the wet lab with great support from other support staff for the labs. And on the dry lab side of things, it's mostly Laura Camila Valera and Dr. Jesse Shapiro who run that section with support from other professors as well. Because I'm on the wet lab side of things, that's where my focus will be for the remainder of the talk. Well, as I mentioned, the wet lab course at McGill is in the fall semester, and it takes place over about 12 or 13 weeks, depending on the year. And the host that we use is a soil bacterium called Arthobacter globiformis, or aglob, as the students like to call it. And students will typically screen anywhere from 3 to 6 soil or other environmental samples before they end up finding a phage. Now, some students get lucky, of course, and they come back, their first soil sample that they bring back has a phage against aglob, and very exciting, of course, but others need a bit more time, or they go out and get more samples. And similar to the Tiny Earth program, we've scheduled the course in such a way where there is that flexibility for you to still be collecting samples while someone else might be already, working on purifying their phage or something like that. So over the course of 13 weeks, students are usually at different places, but by the end, they're all hopefully at the same stage. And this is an overview of the kind of experimental pipeline that the students go through. So first, they're working on just isolating a phage or seeing if their samples have phages. So a lot of screening is being done. And I should mention that 90%, maybe 70% of the labs involve plaque assays. So most of the time, students are just doing plaque assays, whether it's to screen for phages or then to purify the phage that they've discovered and we want to focus on a bit more, or amplify to get enough titer to work with, enough of a concentration to work with. And then once they have their very high concentration lysate that they've been working for 2 months perhaps to isolate and grow up, they actually go down to the electron microscope in collaboration with Dr. Ortega from the anatomy department and are able to look at their phages themselves and prepare the EM grids and get a selfie of their phage that they print and put on their wall at home, as some have done, or just put on their phone as a screensaver, which is in their lab report at the end of the term. But also they will extract DNA from their phage lysate, and they'll characterize it, at least in the wet lab setting, using restriction enzyme digest. So we have a control phage that we've been using all throughout the semester, just for control and comparison purposes. So they'll compare against that to see if they've isolated the same phage, which highly unlikely given the diversity, but also to compare with their peers to see, if you went to the same park I went to, did we get the same phage from the same park, for example. So here are just some photos of student plaque assays from this past fall semester. One sample did not have a lot of phage, not a lot of plaques on this plaque assay. Another was quite dense, so they had a very rich sample in terms of phage. And here's the electron microscope that the students go down to, and actually, well, they make their grids just outside this room, but then they go in with their grids and Ortega here to actually do the imaging, save their selfies onto their phage selfies onto the USB. And here's one example. So students are typically very excited when they get to this point at the end of the term, because they worked, so hard over the past 10 to 13 weeks, isolating something, and it's there because you do the plaque assays and you see the plaques and it's infectious, but you don't really see it until this point. So seeing is believing, and here it is. Now I want to focus on a couple assessments that we have in the course, and of course we have the typical assessments like a final report, and of course the drafts and outlines of that, the different benchmarks throughout the semester for their experimental work, and participation in lab notebooks and things like that. But two that the students really like and that we enjoy as well as instructors are the journal club and phage topic oral presentations that collectively account for a quarter of their final grade. So there's a big focus, of course, on the writing aspect of scientific communication and learning, but also an oral component as well. In the journal club, as I'm sure you're familiar with, is a classic journal club assignment where the students are assigned papers in their groups to kind of present back to the class. And this is in a 12-minute oral presentation format. And we purposely choose papers, it's very blurry here, but we choose papers that cover a wide variety of topics in phage research. So whether it's recent work on phage therapy and phages being used to improve patients' lives. And actually, as a fun fact, the phages in this paper came from the SEA-PHAGES program, which is really exciting. So students get excited about that too, or just phages in the gut microbiome, phage ecology and coevolution with their bacterial hosts in vitro, or other kind of exciting topics. We try and keep the literature recent so that they can see what is actively going on in this field and how their work could contribute to kind of phage research overall. So they really enjoy this assignment. And another assignment that we really enjoy as instructors, but the students also enjoy, is the phage topic presentation, where we put up this big list of about 25 topics, all across different themes of phage research, whether it's phage genetics, ecology, phage host interactions, phage defense mechanisms, or phage therapy. And students select. Oh, here's a photo of a student presenting, but students select a presentation or a topic that interests them, and they really go out and find the literature, compile it together, make it into a cohesive story that they then present back to the class in a 10-minute lecture style presentation with PowerPoint slides usually, with PowerPoint slides. And they also give peer feedback to each other, so by the end of the semester, they can get this feedback and implement it into their next presentations and other courses. And it's really great because sometimes as instructors, we're not fully up to date on a certain aspect of phage research, so we get to include those as well. Like I don't know a lot about arthrophages, so we notice that's one of the topics in the talk, so I learned a lot that day. But it's fun for them as well because they're not all presenting the same thing. And prior to this assignment, we had a final presentation where the students would just present what they did in the semester, which was great for the first 5, but after you get to 20 or 25, they're all doing the same thing, more or less. So it becomes a bit redundant, and for them too, it was becoming a bit kind of tiring and redundant to hear the same thing over and over. So we decided to change it up. They're each talking about something different and actually teaching it back to us, hopefully reinforcing their learnings. Outside of the assignments, we have some optional activities that the students can partake in just to, again, reinforce their learning and keep them interested in the course. So one that we started up this past year was a phage modeling play activity. So one day after lab, if students had time, if they were interested, totally optional, no grades, no participation. They could stick around, and I brought this modeling clay in different colors, as you can see. And students were kind of told to make a phage, but keep in mind, the different colors might represent different genetic diversity, right? And we spoke about how diverse phages are in terms of their size, genome length, genome characteristics. So try to use different colors, try to use different shapes in terms of the phages that or try to make different morphotypes of phages. So we spoke about the different morphotypes and things like that. And yeah, by the end, we had maybe 10 to 15 students who stuck around that day, who made phage keychains, phage earrings, just phages of all shapes and sizes that they then got to keep. So, fun activity that they enjoyed, and there were some snacks as well. There's on the last day of class a phage trivia game as well, that one student even baked phage cookies. That really was delicious, but it looked so great. And yeah, the phage trivia would cover topics that were either presented as part of their topic presentations to, again, kind of remind them of some of the take-home messages of these presentations, or just other facts that we've briefly mentioned or want them to know just going forward about phage, quite honestly. And at the end, I don't have a photo, but they could take home some 3D printed phages if they're group one. That's very competitive. At the end of the semester, in terms of feedback, McGill runs, I guess like other institutions, a course evaluation system that's only released back to instructors once the final grades are submitted. So some feedback that we got from students over the years has been really positive. One student said they never enjoyed coming to class every Tuesday and Thursday as much as they did this semester, and they enjoyed the work so much. Great sense of personal pride when they could finally see their phage on the electron microscope. They got really involved in the scientific process, which they really enjoyed. These are all from different students, and this last student found it incredibly engaging and a great way to get comfortable in the lab, trying all kinds of different techniques, which, I mean, I've never done EM as an undergrad, so it's a really great experience. I think that one especially to be able to do and at least see an electron microscope, like centimeters of where you are. So with that, I'll end it there. I think there's some time for questions, and I just want to thank, of course, Jasmine and Corinne and Laura for their support in teaching or in workshops, the funding, the SEA-PHAGES students who are in Canada this year, because there were quite a few, and all of you. And if you have any questions and I'm not around or you can't find me, of course, we can chat during the break. But feel free to reach out by email or on Twitter. And if you're curious about starting SEA-PHAGES at your institution, I'm happy to chat about that as well. Thank you.
They found phages for Arthrobacter in a sample. Does that mean there's Arthrobacter in the sample? Because phages are very specific.
Yeah, phages are typically very specific to a species or even a strain. Now we're finding more and more some phages that can maybe cross certain like genus boundaries even, but for the most part, yeah, if they're finding an Arthrobacter phage, it's because there's arthropods in that nearby.
How far do they go into characterizing the host range of these phages? Do you test different, like do you test the host range at all, or?
It depends on the year. So some years we have more free time than others, just based on how the students progress over the term. And those years where we do have the extra time, we do bring in other hosts. Some that we know it won't infect, like there's a very low chance that an Arthrobacter phage will infect E. coli, for example, a lab safe strain of E. coli, but just to show them that like, hey, it's not infecting and it's a good negative. And some other soil bacteria as well. So just trying they can do these assays.
Fantastic. Just wondering if this deals with residents.
This
The courses that like workshops.
the workshops.
Yeah.
Yeah, I don't see why not. I mean, at the time when we developed them and we ran them, it was specifically targeted for undergrads, but they're still in this very structured learning environment. So having this optional they could take that would, yeah, that's you're interested in, but yeah, I think it could be, anyone could perhaps register for that, and we could adapt the curriculum for them.
Unfortunately, interested.
Okay. I'm happy to share also like any of the material to slide.
Okay, so I have a question about the the Galaxy interface.
Yes.
The bioinformatics curriculum. So did you get any student feedback on interacting, and how did that look for them currently working? Like, were they kind of given a step, or was it a general protocol for assembly that you need buttons for either, or?
Yeah, so we would go through it together. I would kind of like this, I'd project my screen and then they'd have their computers open. And they could follow along with their own files. So I can upload the file, I had to run the tool with the inputs and outputs. So some of the feedback we got from the first set of workshops with the command line sessions were that it felt a bit rushed because they were learning this whole new command line environment in a crash course setting, but then have to immediately apply it. So that's the whole factor.
So you didn't have enough time to do a bash primer or anything like that?
No, so that's why, because the library prep steps took longer than the other.
So just a small follow-up on, with the Galaxy interface that implemented, were there any priority points for you as instructors to make sure that students kind of understood what the steps were doing, or what, in terms of like, this is assembly, this is the quality check?
Yeah, so I didn't show here, but we've had slides for all of the different tools showing like how they and how they work, what they're doing. And yeah, this is what students have in the report.
Do most students have at least one course in molecular evolution of some kind? You know,
I think that would
a
be the
nucleic acid analysis based course, not like a hard course, but something where they see a little bit of these are things that you would be on.
Yeah, I think like in the genetics course, the intro to genetics course, they would learn about sequencing there and some of its applications. But not necessarily the tools or ways that you do these applications, just that it's done. So
Thank you.
yes.
On the slide that you have up right now, you have the Digital Research Alliance of Canada.
Yes.
And wondering, you talked about when you set up this Galaxy server, it sounded like that was sort of a departmental asset or a lab group asset. Is that the case? Or did you use the draft as the source of that computing environment?
Yeah, so the Digital Research Alliance of Canada, or formerly Compute Canada, we used, it was a cloud VM that we used, that virtual machine for the bash session, like in the first set of workshops. And then also we set up a Galaxy server on the cloud VM.
And are you giving them feedback about this so that? So I know from interacting with them,
Yeah.
the use of their cloud resources, especially in teaching, they are looking for any example of it because it's a huge added benefit of the resources that they have. So when they have to justify it back to government, it's usually opportunistic, but it's actually widely impactful. So
Okay, yeah, I can reach out.
please make sure they don't make it.
Thank you.
I'd say the floor is open for about 5 more minutes for questions for either of the keynotes at this point.
Good for an either. When there's an opportunity to publish results from like student work, is there opportunity for them to be more involved with that? Like, are they offered opportunities to write or contribute in a meaningful way that way?
So we're kind of incorporating in baby steps, and that was always a long-term goal. And so this year, what we're doing is we're going to have students do direct studies with us who did it last year to publish in our local Carleton University undergrad science journal.
Sure.
And start there. But we're trying to we love the poster, and so we were thinking of maybe offering a choice that they could try to write it up and submit it to our local journal again. But we really like the poster presentation, and I don't know, so we're